The challenges were just the usual, as you know, of asking a question and hoping that you will get some insight, whether it gives you the answer that you think it should give you or something completely different. It's the asking of questions in a most, I think, open-minded way. I think I'm very much a hypothesis-driven research person. Nevertheless, I am also a staunch believer that what you have to do when you have a hypothesis is try to disprove it, not to prove it. So I think the challenge was, of course, doing good experiments that would give some sort of clear answer, yes or no, that would take you to the next step. And sometimes you get fuzzy answers. So that was the usual frustrations. The lab took off fairly fast because there were clear directions we needed to look into and walk into. And I was joined by a fantastic crew of students, too, right away. Postdocs very soon. Wonderful technical support from the first day, before I arrived, actually, Beth McLeod. So we were equipped to get going and then take the different questions, whether it was really what is Oskar doing as an RNA, what does it encode? We knew it made a protein, what does that protein do? That took years, if not decades, to get insight into, actually. But really understanding more about the transport process, what part of the RNA is responsible for its getting localized, what is the mechanism of localizing? Is it that the RNA gets sort of swirled around and then captured? Or is it, there were two other prevailing models, and all three pertain in biology. RNA being uniformly distributed in a cell and then being just degraded throughout the cell but protected at one location. Or other possibility is that it really is a question of active transport, like components of vesicle, or like vesicles and membranes in cells undergoing vesicle transport. Could RNAs be also moved by molecular motors, mechanoenzymes? And though these models existed, we didn't actually know at the time. It's the ideas that people were having in the lab. Very often I myself had a gut feeling, and it's dangerous to say go with your gut, but there is an element of that. When you know a system, or you've been living, or it's your brother, Oskar, or whatever, for a long time, you sense, when things aren't quite right, that maybe it could be this way, and sometimes it can take you completely down the wrong path. But really discussing those sort of gut feelings or ideas, you could say, vague ideas, was, I think, very important, because it really did lead us to things that were completely unexpected and turned out to be very useful. The other thing is really, I think, that really encouraging people in the lab, they had their project. I really don't like when I have a project. We're doing science is what we're doing. We're asking questions, and the general question is this. Is it a project? For me, a project is when you're going to build a lamp or something like this. You put the thing together. That's not how you do science.
Because you don't have extraction of what is going to...
Or there's a problem if you do. But it's the importance, while you're, on the one hand, doing your experiments and you observe something and you continue, but it's also tinkering. If you have the time, and especially if you're a postdoc, by the time you're a postdoc, you have your driver's license, essentially, to do, research. And on the side, not at great expense, obviously, but to try little things, really little risky, crazy things, because you just really, nature holds many more secrets than we know. We know so little. And by uncovering a stone that you don't think really should be on your path, and you take a sidestep, suddenly you realize it's part of your business as well. So I think that really, not being too...
Yeah, no, and I guess this has been also a strong part of the EMBL culture, because, there's been a lot of, examples of, quite big I would say, breakthrough coming out of the EMBL due to serendipity.
Absolutely,
Coming
absolutely.
back a bit to what we were discussing before,
Yeah.
but is absolutely another.
Absolutely. People have said, you're still working on Oskar? It's embarrassing, right? But it served as, it's been a Swiss Army knife for studying post-transcriptional RNA-based regulation of development. Every single process of RNA regulation somehow has impacted Oskar and also how it's dealt with in the cell. I think that there are things that have been learned from that about also how Oskar is transported, and it's work from our group. The most recent work, a paper last year with Simon on the switching from dynein to kinesin, that I think is very pertinent in some large cells like neurons, etc. And of course, Oskar itself, Cassandra Extavour found long ago that Oskar was not in the germline of crickets, but was in the brain.
Yeah.
So I think there's space for that. No, I think that Oskar has served for a very good model for understanding how an RNA can be regulated in certain contexts, or indeed, you know, how RNAs are packaged.
I mean, the other thing that I always found in perspective is how difficult it is to deal with RNAs rather than to deal with proteins somehow. And, you know, I still feel, you know, one of the first impressions that I had coming to the lab is, you know, how little we knew and how little tools we had to actually try to probe the structures of RNAs that were important with interacting with proteins. Remember, I mean, you had to do all the molecular biology chopping in pieces without really knowing what would be the result. It's just like stumbling around a dark room to try and find the right piece. And
seeing a predicted stem loop with the lousy programs there were, and then seeing if it was a real one by making mutations and then compensatory mutations, which really did.
Here, we are coming now to the liquid particles, and I still have the feeling that there will be years and years of
and to AlphaFold, and we can talk about AlphaFold, about new methodologies, right?
Yeah.
So you know, to this day, RNA folding remains a complex problem.
So we'll look forward to another, you know, years of discoveries there. I mean,
I think that really that's where the AI and experimentation and back and forth with computational biology is really going to, I mean, the breakthroughs with AlphaFold are just unbelievable.
Yeah, no, and this is for proteins that we understand very well. So,
absolutely,
you know, let's see what happens with the RNA.
I think it's going to go far, and
it's quite a challenge.
It's moving, absolutely.